Synapses between neurons separate postsynaptic dendritic protrusions, spines, from presynaptic axonal boutons. The spine head is largely composed of actin. The postsynaptic membrane, or PSD, is packed with glutamate receptors. Synaptic activity tags the stimulated spine, red asterisk, so that it responds more strongly to further stimuli. The stimulated spine increases size. The calcium calmodulin-dependent kinase 2, CAMK2, is a dodecameric enzyme abundant in dendritic spines. The inactive form binds both G and F actin. This study investigates these interactions in an in vitro system that overcomes in vivo complexity and size limitations. Turf microscopy revealed GFP-TAT CAMK2 holoenzymes attached more stably to junctions versus non-junctional locations in rhodamine phalloid and F-actin networks. Multiple filament junctions were predominantly formed by single holoenzymes visualized by electron microscopy. Associations at non-junctional locations were comparable to the weak G-actin association. We studied 3D actin networks cross-linked with CAMK2 from 3 different species. All formed random networks at physiological molar ratios, stable over tens of minutes as documented by centroid tracking and nearest neighbor analysis. Upon stimulation, the NMDA-mediated calcium influx changes spine volume in 2 phases. First, the spine expands to twice its basal volume due to actin polymerization, initiated by CAMK2 activation and CAMK2 actin dissociation. In vitro, the dissociation from F-actin was coupled to network disassembly. Second, the spine compacts to a stable volume 1.5 times the basal volume. What drives compaction? Aggregation by crowding agents was too slow, while, even though myosin II is present in spines, the CAMK2 F-actin networks resisted motor forces. However, myosin minifilaments rapidly compacted disassembled networks to resolve the conundrum. This schematic puts our data in context of the physiology. The strong multivalent actin avidity of junction-localized CAMK2 fixes spine size over hours for measurement of the volume difference between stimulated and resting spines. The fast calcium calmodulin switch, based on the weak monovalent affinity, drives remodeling. In expansion, the increased G-actin pool stimulates polymerization. In compaction, network disassembly unblocks myosin activity before incoming CAMK2 reforms the network.